50% of information stored within the brain is associated to our vision. This is because the visual cortex corresponds to around 55% of the entire cortical area of the brain compared to other sensory systems. Vision is required in order for us to perceive the world around us, allowing the images we see to have colour, shape, depth and movement. The visual system involves pathways for neurons to receive and process visual information that project to the structures of the visual pathways beginning at the eye. Visual information is then conveyed all the way to the primary visual cortex, which is responsible for processing and interpreting images. Other important structures involved in vision are the optic nerves, the optic chiasm, the optic tracts, lateral geniculate nucleus within the thalamus, and the optic radiations. The retina is situated here at the very back of the eyeball. Its purpose is to receive light which the lens has already focused and then convert this light into neural signals, which will then be sent to the brain for visual recognition. The light enters here through the cornea, then it passes through the aqueous fluid at this anterior chamber, and next it passes through the lens and onto the retina. Once the light hits the retina, it travels through each of these cellular layers until it reaches the outer parts of these specialized cells here known as photoreceptors. There are two types, rods and cones. Cones allow us to perceive colour under normal lighting conditions, whereas rods allow us to see in dim light but do not allow for the perception of colour. It is at these photoreceptors where the light interacts with a molecule called photopigment. This interaction starts a chain of events which serves to propagate the visual signal. Signals are sent to the brain via the bipolar and ganglion cells, whose axons form a nerve fibre layer on the inner surface of the retina as shown here. This layer forms the optic nerve and exits through the back of the eye. The optic nerve then carries visual information toward the brain via the thalamus to be processed. These horizontal and amacrine cells allow for sideways integration of retinal activity across areas of the retina.
The optic nerve is part of central nervous system, which also called the second cranial nerve. The job of this paired nerve is to transfer visual information from the retina to the vision center of the brain via electrical impulses. The nerve is surrounded by three layers: the tough outer dura layers, the arachnoid, and the vascular perimeter. The optic nerve leaves the optal cavity through the optic canal and unite with the optic nerve of the opposite side to form the optic chiasm. The optic chiasm is located at the junction of the anterior wall and floor of the third ventricle. The optic nerve fibers on the nasal side of each retina cross over to the opposite side of the brain. Meanwhile, the temporal fibers just stay at the same position. The crossing over of optic nerve fibers allows the visual cortex to receive the same hemispheric visual field from both eyes.
Following the crossing of the fibers at the optic chiasm, the now unified left and right visual fields travel down their respective optic tracts before travelling laterally around the midbrain to reach the lateral geniculate nucleus in the thalamus, where the majority of fibers synapse with the neurons of the LGN, whilst the remaining fibers travel either to the superior colliculate nucleus or to the pretectum. The LGN consists of 6 layers of neurons, the first 4 layers being composed of parvocellular neurons that receive information from the P cells in the retina, and the final 2 inferior layers being composed of magnocellular neurons that receive information from the retinal M cells. The layers receive information from one eye, and in the case of the parvocellular layers, one visual field, which results in the preservation of separation of visual fields. Parvocellular cells make up the parvocellular layers of the LGN. They are relatively small compared to M cells and are responsible for the analysis of shape, size and colour, as well as spatial resolution and visual acuity. Meanwhile, magnocellular cells make up the magnocellular layers of the LGN. They are relatively large cells that specialise in the detection of the location, speed and direction of a moving object. Whilst the LGN's role is to filter visual information and then be relayed to the visual cortex, the superior colliculus's role is a little different. The superior colliculus receives information from both the eyes and other sensory data from the body, which it uses to control the orientation of the head, the movement of the eyes, aiding in the tracking of moving objects, and additionally, the superior colliculus plays a role in decreasing the general distractibility of an individual and is important for visual focus. Axons leaving the LGN travel further into the brain, spreading out to form the optic radiations and moving slightly superior to and lateral to the temporal horn before moving back towards the visual cortex again. Unlike the optic tract and nerves, the optic radiations actually spread out a lot more, so whilst the superior portion of the radiations travel over the temporal horn and pass under the parietal lobe, the inferior portion of the radiations travel outwards around the horn, passing through the temporal lobe, forming Meyer's loop. The radiations then enter the primary visual cortex in the occipital lobe.
As mentioned previously, the optic radiations terminate on the visual cortex or the striate cortex, being Brodmann's area 17. The visual cortex is located on the superior and inferior portions of the calcarine fissure and is important for combining and analysing visual information. The superior optic radiations come into the striate cortex and terminate on the cuneus gyrus, and the inferior radiations project to the lingual gyrus. The cortex is comprised of horizontal and vertical columns, and their cells will correlate to a point in the visual field. The vertical columns are also organised into alternating ocular dominance columns. The magnocellular areas of the cortex is associated with motor stimulation, and the parvocellular is important for colour. The information is then projected to Brodmann's area 18 and 19, which is the extrastriate cortex or the visual association cortex. Various cortical areas are involved which aid in visual processing, such as the ventral pathway that deals with fine detail, which is used for identifying objects, whilst the dorsal pathway is responsible for identifying motion and dealing with spatial location.